Good morning, everybody. Quick view of Sentinel Dog. She's watching to make sure that the old lady across the road doesn't come out and get her trash cans. If she does, she'll start barking at her for some reason. The only person that she ever barks at. It's a complete mystery. Okay, so before we get going, a couple of announcements. One, which you should have seen already, exam has been delayed or pushed back by one class period next week. So the exam will be on, crikey, I've got a lot of documents open. There you go, this is the one. It will now be on, wrong exam, Thursday the 14th of October. Same deal as before, it'll be open from 9:30 till midnight, 9:30 in the morning, that is. I'll be in Zoom at 9:30, probably for, I don't know, half an hour, something like that, if you need any help. And I'll be available by email for the rest of the day. Now, that has caused a couple of like ripple-on effects, which are moving homework 7 and 8 a week later. Otherwise, you might not have actually had the stuff that's in the homeworks. There may be problems with homeworks 5 and 6. Right, I haven't had time to go back through them and double check that everything that's in them we will have covered already in class. So if you have any problems with that, just let me know and I'll fix that. Second announcement is, this is more of an issue for my lab on Tuesday, but we didn't have time to actually talk about, where are we at? Which one is it? Oh, that one. We didn't have time to talk about the standard curve gel electrophoresis stuff. So I have made a short equivalent, essentially, in my office called Standard Curve Lab. So please refer to that so you get an idea of what you're supposed to be doing. Anybody else can watch it as well if you want a recap or whatever. It's for everybody, but specifically for the Tuesday group, because it was yeah, probably best not to go into more details than that about that lab. Okay, so I think that's pretty much it. That means we have a much more relaxing time of it. Oh, craps, that's what I meant to do. Also, it came up in the lecture yesterday. Someone reminded me that there's a really cool video on DNA replication, particularly of the lagging strand. So I've put that in there. Let me go grab the link. Come on. It's actually a real cool video because it's got a whole bunch of different bits and pieces in it, including, I don't know if you remember, I don't know if it was this class or another one, but I spent ages trying to find a really cool video of cell division. There we go. Yeah, come on. There we go. And I just really like this because it's a real-time in vitro look at, you know, it must be mitosis would be my guess. So here are the compacted, condensed chromosomes, and they're jiggling around, lining up. Oh, crap's happened already. On the metaphase plate. See, it doesn't take very long. I think it's sped up a little bit. Right, so here's the metaphase plate here, and you're going to get the two daughter cells here and here, right? The plane of division is going to be vertical. And so you see them getting pulled apart, and then as cytokinesis occurs, these chromosomes will decondense into just an amorphous lump of DNA. right? Which will then essentially be the cytoplasm, the nucleus, sorry. So this is what I really wanted to show. So it's really hard to describe how the lagging strand works, right, with words, because it's just a complex process that kind of requires you turning your head around a little bit. And so this is I call it a molecular animation. And essentially what it is, you know, everything that they knew at the time they made it about what this looks like is how they portray it, right? So obviously, you know, there's lots of space around it, which there normally wouldn't be, to make it clearer. But these are the enzymes that are being used, and this is what it actually, as best as they understand it, looks like. And so essentially down, let me blow this up a bit. This strand down here is the leading strand. You'll see that it gets synthesized continuously. These blobs are enzyme complexes. Pretty much anything that does anything in biology, or certainly the biology that we're learning, is going to be a complex of proteins, right? So not one protein, many proteins, sometimes tens of proteins, all kind of glob together in a particular way to perform a particular function. So this glob over here is, well, both of them, they kind of look like pom-poms, are the DNA polymerase complexes, right, which are synthesized in DNA. This is not differentiated between eukaryotes and prokaryotes, right? Essentially the same principles apply. We also have a enzyme at the front. This is helicase. It doesn't show topoisomerase or DNA gyrase, for simplicity's sake, I imagine. But this is helicase unwinding. This is the leading strand getting synthesized continuously. And this is the lagging strand. So what you can see is that replication fork is going left a ways, right? It's going into the kind of left of the screen, right? And so the leading strand is being synthesized continuously, right? Because the 3' end of the leading strand is about here somewhere inside the DNA polymerase. Now the 3' end, if this was left going out, the 3' end of the newly synthesized lagging strand would be going that way, right? Because it's an anti-parallel double helix. So what happens is that the template for the lagging strand, or the parental strand, is looped back so that it can also be made in the same direction as the replication fork. However, the problem is, and you'll see it in this animation, which is why it's such a fabulous visualization of this. When you do that, you will hit what you have already done, right? Because you're essentially synthesizing the strand backwards in a way, right? So let's see what that looks like. So if you look carefully, here's the already synthesized bit, gets kicked out, looped back again, and you'll see the bit that was just done. Ah, dang it, it's just too fast. Oh, hey, I remember. Hang on. Yeah, this is more like it. One of my students actually taught me this many moons ago, because I was always going, shit, it's gone. Come find it. I'll actually have to watch the rest of this. It's got some other stuff that I haven't seen yet. So here's the bit of the lagging strand, Okazaki fragment that's been done before, right? Completes that, loops back again, and you'll see the bit that has just been released. It's up here. It will start coming in and then kicked out. And again, that bit that has just been made, you'll see it come back again. There you go. And then kicked out, right? So that is why the lagging strand is synthesized in chunks, the Okazaki fragments. Because by looping the DNA back around, that allows you to have this whole complex, right? Moving as one, synthesizing both strands in the same direction, even though one of the strands is going in the wrong direction, right? Running templates. But that creates a problem that you'll then basically run into what you've already done periodically. So that's why the lagging strand is constantly being released, loop back in, synthesize a bit until you hit what you've done. Then it gets released out again. So here's a newly synthesized Okazaki fragment here. It's about to hit one that's already been made, and it kicks out. All the while, the leading strand is being synthesized continuously. So that's a real cool visualization of what's actually going on. Certainly, hopefully, makes more sense than me just waving my hands around and using words to try and describe it. Actually, what else cool is there? I think these are just fabulous. I'm a bit of a nerd like this, I guess, but so this is the helicase, right? And so you actually see this kind of almost spinning. We have other bits and pieces in here. We have primase, and we have the newly, there's a primase in there somewhere. It's just fabulous. Isn't that amazing? People who did this are so clever. Anyway, where's the other bits and pieces? Oh, that's a new one. All right, we'll save that for later. Cool. All right, did that help? Hopefully. That I will upload to course content after class. I meant to do after class yesterday, got sidetracked with other stuff. But yeah, that will go up in a wee bit, and I'll almost certainly be using that. Oh, that video again. Also just love imagery. Imagery is such a powerful tool. It really is. Let me show you a cool one. Where did I find that? This is just a fave picture of mine. There you go. So this is an alligator embryo. And I need to go back to the actual source and figure out how they did this. I don't know whether this was done by immunostaining, so labeling different parts, or if it was done using fluorescent reporter genes, which fluoresce different colors under different lights, essentially. And this is a composite image of all of these different colors stacked together. But it's just super, super cool because here's the myocardial tissue, which you can see is starting to develop into a heart. Here we have other muscle tissue. These will develop into the vertebra and different muscles in the back. You can see all these neurons, right? So this is the developing brain, which is kind of small, lizard brain, I guess. But you can see all the different processes of these neurons. Here's a spinal cord essentially developing along here. It's just beautiful, right? So this is taken with a laser scanning, confocal laser scanning microscope, which is super, super neat. I've had fun playing with them in the past. We also tried to get a grant for one here, but they're about $500,000. Yeah, they're seriously, seriously expensive, but allows you to make just the most amazing pictures. Anyway, I just love looking at stuff like this because I think it's super neat. So getting back to business, right, we're going to start with prokaryotes. Not really, because prokaryotes are that much simpler. I mean, the basic principles are the same between both prokaryotes and eukaryotes. It's just, I guess, force of habit, really. Typically talk about prokaryotes first. So in prokaryotes, we have, or actually all organisms, series of phases of DNA replication. We got to start it. We've got to unwind it. That's the job of helicase. You actually have to synthesize it. And then at some point you stop it. Or really, it's not so much stopping it as finishing the job, right? And this will get back to why the lagging strand is called the lagging strand. And so remember what I talked about earlier in terms of relative strength of bonds between A's and T's versus C's and G's. Well, this is one place where it becomes important. And so essentially what happens is we have an origin of replication. One in E. coli, prokaryotes, thousands upon thousands in eukaryotes. And essentially these origins of replication, or this origin of replication, is recognized by a bunch of proteins. So recognized based on its sequence, typically, as well as in part by its shape. And what these initiator proteins do is they bend the DNA, right? And so my favorite analogy, so I was talking about yesterday, braids, just because our exchange student wears braids occasionally. So whichever analogy works for you is great. Either talk about a rope or talking about like your younger sister's braids. Doesn't matter which. So if you get a rope piece of twine and you twist it in the opposite direction to, I should just go and get some from the backyard, really, forgot. Twist it in the opposite direction to its natural twist and it will open. So next time you find a piece of twine or rope or whatever, or a braid, ideally of a family member, not some random person in the street or on the bus, twist it in the other direction to that which it is wound and it will open. And that's essentially what the initiator proteins do. They force the DNA to bend in a particular way. And actually this is how RNA polymerase works as well. And in doing so, those weaker bonds between the A's and the T's allow the DNA essentially to go from being twisted to being popped open. Right, that's the very, very first step. Now the problem is it wants to go back again, right? Because its natural lowest energy state is the double helix, right? And so to prevent that from happening, these proteins called single strand binding proteins, which is kind of self-explanatory what they bind to the single stranded DNA in that little bubble, and they stop it from twisting back and base pairing again. Literally physically stop it, right? So the bases that are in here are not actually able to come close enough to form those hydrogen bonds again. So that holds that bubble open. And that allows helicase, our unwinding protein, to get in there. That's the, where is it? That's this blue protein here. Obviously, proteins don't have colors, but this is the protein at the very tip of the replication fork. And essentially, as soon as that gets in there, it's going to start working to expand that bubble, right? Because there has to be sufficient space for all of the other stuff, this replication machinery, to actually get in there. As you can see, they're big things, right? These are big protein complexes that occupy, in molecular terms at least, a bunch of space, right? Okay, but that gets back and that creates another problem, right? And so I actually like thinking about DNA replication in these terms. Essentially, you solve one problem and you create another one. It's like, oh, again, and you fix that problem and you create another one. It's just a whole bunch of problem solving or creating. So going back to our analogy of the rope or younger sister's braids, right? And you can do this as an experiment, like I said before. Now, nail one end of that to a wall. Probably best doing it with rope rather than your sister, because your parents might object to that. Or your sister might, actually. Or even your brother, if your brother has braids. I don't know. Anyway, whatever. Now stick that pencil in there and push it, right, down the length of the rope towards the fixed end. So you open up the helix, right, of that rope. What's going to happen ahead of the pencil? If the other end that you're moving the pencil to is fixed in place, what's going to happen ahead? If you unwind the rope behind the pencil, what's going to happen ahead of the pencil? If I had time and I knew where it was, I know it's out in the garden somewhere, I just don't know where, I'd go and grab some and I'd show you this. It's actually really cool. I recommend you do it yourself. What's going to happen? If you have a series, of strands twined together, right, in their lowest energy state, yeah, Cynthia, it'll bunch up. Because essentially, when you reduce coiling in one place, you increase it in another place. So as helicase is opening up DNA, reducing coiling, essentially, is increasing the coiling of the DNA ahead of the helicase or the replication fork, right? And in this case, it's ahead of the replication fork. This extra coiling actually has a name. It's called supercoiling. Essentially, if it goes on for long enough, you will get DNA will coil on itself, and those coils will coil on itself, and you'll just get a coiled lump of DNA, which you can do nothing with. Now, if that DNA had a free end, say you're right at the end of a eukaryotic chromosome, maybe, what you'd actually see is the end would spin, which you can do as well. If you just get a piece of twine, stick a pencil in it and move it along without holding the other end, that other end would spin, right? Because it's relieving that, coiling essentially that's trying to be introduced by reducing it elsewhere. And so that creates a problem, right? Because DNA isn't free to rotate like that in the cell. It's fixed, either because it's circular in prokaryotes, or because it's one of many replication forks in a long linear eukaryotic chromosome. So we solved that problem with proteins called topoisomerases. An example of that is DNA gyrase. That's what it's called in prokaryotes. So their job is to move ahead of the replication fork and relieve the supercoiling, right? That is building up. I don't actually know how they detect that. Maybe just the change in the shape or the tension in the DNA would be my guess. Essentially what they do, if you imagine the DNA double helix like so, right? The topoisomerase is actually kind of neat in many ways, because it will break the DNA, either one strand or two. And so it's kind of easier to visualize with a double-stranded break. Normally, this is a catastrophic event, right? This is what gamma radiation does to your DNA. And why, if you jump in like the spent fuel rod pool of a nuclear reactor, sorry, you'll end up a, dead soupy mess afterwards, right? Because gamma radiation breaks your DNA physically, breaks it apart, right? And so this is what the topoisomerase is doing, but it doesn't cause problems, it solves them, because it will cut or break the DNA apart, hold on to both ends. And now you have a free end. And essentially what will happen is the DNA will rotate to relieve that supercoiling. And then the topoisomerase, actually, you've got to do it twice. I can't bend my arm that much around, so you just have to imagine, right? Because obviously you want the same end stuck to the same end, right? You don't want to like swap over a chromosome. That'd be kind of weird and wrong, or the strands rather, right? So, but by making that free end, it allows the DNA to swivel, and then it sticks it back together again, and moves along and does it again and again and again and again. Or another way of doing it, and this depends on the type of topoisomerase, is you can actually have a single-stranded break. So if your DNA is like so, you're just going to nick one of the strands, but that essentially has the same effect because, oh, there you go. I could actually do it this time. It allows the DNA to rotate around, and then that strand gets stuck back together again. So they're actually really cool. You can actually even use them in molecular biology. You can use them for cloning genes and stuff like that. I haven't done that for many, many, many years. But anyway, so that solves one of the problems, right? The problem of supercoiling. So topoisomerases are all about that. Okay, but as you probably imagine, we're not done with the problems. So let's go back up here. Come on. Okay. There we go. All right. So what did we learn over and over and over and over again? Last lecture about where new nucleotides are added to. What carbon and what attached to that carbon does a new nucleotide get attached to when we're synthesizing DNA or RNA? Which carbon, which number, and what's attached to that carbon? Don't all answer at once. My dad's favorite saying. Okay, go back. It's a big part of how we talk about the direction of DNA, or the directionality of DNA. It has a something end and a something end. Numbers. Yeah, 3'. Thank you, Cynthia. I was starting to talk about how many fingers do you have on one hand and stuff like that, right? So you saved me from that. Right, so it's the 3' carbon. What's attached to the 3' carbon that is required for making a new phosphodiester bond? Yeah, I'm guessing that's like an OH as in a hydroxyl rather than an O. Like
yeah, my bad.
Right, you need a 3' OH. I was assuming that it was a hydroxyl group. Bit better to type it like that though, just to avoid confusion. Okay, so you need a 3' OH, right? That's the only way you're going to stick a new DNA nucleotide on, right? DNA polymerase requires it, absolute requirement. No 3' OH, no new DNA. Okay, where in here do you have a 3' OH hanging around? Or blow up super, super big. do you see a free spare 3' OH anywhere in here? Yeah. Give you a hint, it's a trick question. Well, not a trick question, but you know what I mean. Do you see one? Well, where's the daughter strand though, Cynthia? We haven't started synthesizing DNA yet. We've just opened things up. So both of these strands here, these single-stranded DNA, both are template strands. Good idea though. But yeah, this DNA replication has not started yet. Well, I mean, we've initiated it, but we haven't actually started making DNA. So where's that 3' OH that is so essential for adding new nucleotides to? It isn't there. And so that's a big problem because DNA polymerase will look at that and go, nope, I'm not touching that, and go elsewhere, do whatever else DNA polymerase does. So that's a big issue, right? That's another problem. So we need to create a free 3' end to begin DNA synthesis, but DNA polymerase won't add a nucleotide to a single-stranded bit of DNA without a 3' end. Right. So how do we solve that problem? We solve it using a different enzyme called primase. Now, as you can imagine, well, primase is an RNA polymerase. What do we use in transcription to make mRNA? RNA polymerase. Does RNA polymerase care about there not being a 3' OH to add its RNA to that bit of DNA? No, it doesn't, because otherwise you'd never get transcription. It's quite happy adding an RNA, going, oh, that looks good, junk, stick that down there, right? Quite different from, at least in prokaryotes, DNA polymerase. So the RNA polymerase called primase comes in and makes a short piece of RNA. I can't remember exactly, about 15 to 20. Oops. Or the single strand, sorry. I guess trying to type while I'm slouched on the couch isn't a very good plan. And so once that short little bit of RNA has been made, right? So now we have basically an RNA DNA hybrid, right? Or mix. Now we have a 3' OH. It doesn't matter that it's a 3' OH on an RNA nucleotide. That's irrelevant. As long as there's a 3' OH there, DNA polymerase will go and go, okay, I can deal with that. And it will begin synthesizing DNA. You can actually see the 3' OH here, kind of hanging around, waiting for a new nucleotide to come in. So this RNA primer has to be made every single time you initiate replication. So let's go back up here. So how many times per replication fork will primase need to make a primer for the leading strand? How many primers do you need for the leading strand in a replication fork? And the DNA is synthesized continuously, or the daughter strand is at least. How many RNA primers will you need for the leading strand? Yeah, good job, just one. How many are you going to need for the lagging strand? I mean, just look at the picture here. Yeah, you're going to need several, or in the case of eukaryotes, thousands, right? You need one primer or RNA primer for every Okazaki fragment. And we'll get into the problems that this causes later on. So every single time that RNA polymerase loops out, it comes back again, you're going to have to make a fresh primer for it to continue synthesizing that new Okazaki fragment. Okay, anybody got any questions so far? Yeah. Cool. All right. So this is basically kind of a recap of what we just talked about, but it's a nice visualization of the whole process that has to happen, right? How many different primers that you have to make. And so when elongation occurs, essentially it's just the same picture that I've shown you, just in a different orientation, different format, right? You're going to be adding that nucleotide to the 3' end. And essentially what happens is we have hydrogen bonding, right, which allows the nucleotide, the correct nucleotide, to come in. Sometimes that doesn't work, and we'll talk about how that's fixed later. And then each incoming nucleotide has 3 phosphates on it. And it's the energy in one of those phosphate-phosphate bonds that's used to form this phosphodiester bond. Basically, you rip off the hydrogen from this hydroxyl group and add it to a hydroxyl group off of one of the phosphates, creating water, and in the process, you get a phosphodiester bond. Now, in prokaryotes, they're a little simpler. We have 5 main DNA polymerases. There's quite a few more in eukaryotes. And DNA polymerase 3 does the main job. That's the workhorse of DNA replication in bacteria. The other ones, of those, we have DNA polymerase 1. So its job, which we'll get to in a tick, is to remove these primers and fill in the gap. And then the other RNA polymerases are all involved in DNA repair, primarily. And that's really all about correcting problems in DNA replication, which they do occur, not super frequently, but they do occur fairly frequently, before DNA replication is complete. And that's the same in eukaryotes too. Okay, so again, going back to creating this RNA primer, so that solves one problem, right? It solves the problem of that 3' OH being required for DNA replication. but it creates another one. What kind of problems do you think this will create? What problems do you think will arise from having a piece of RNA or many pieces of RNA in a stretch of newly synthesized DNA? Do you think that RNA will be able to be copied into DNA by DNA polymerase if it was left there next time that cell divides? No, I don't think so, right? It's not going to be able to be template for transcription, right? Because RNA polymerase requires DNA to make a copy of the open reading frame. What else? So we'll get to this when we talk about PCR and lab in a few weeks. But what do you think would happen if you had all of those Okazaki fragments and you try and you separated that DNA, right? Say for transcription or maybe even for DNA replication again. What's going to happen to all those fragments? Are they going to go back in the right way? Are they going to get stuck back together properly? Are they going to reform onto the old strand? The answer is probably no, right? So having that RNA primer there and those fragments particularly is a big, big problem, which we've got to solve. So to finish up DNA replication, we need to do three things. One, remove the RNA primer, right? We're DNA based, our information is stored in DNA, not in DNA and RNA. Second, you've got to fill in the gap, right? Not doing so would be like, repairing a railway line by taking out a bad rail and then just not bothering to replace it, which would not be good. And then the last thing, and this is actually really important too, is you need to seal the backbone, the phosphate sugar backbone, because without doing that, essentially you have a break in the DNA strand, which will cause all kinds of problems later on. And that's essentially what needs to happen. Now, this has to happen for every primer. Now, think how many primers are in the lagging strand versus the leading strand. It's going to take a lot longer to do all this for the lagging strand. So, and this is actually an area that prokaryotes and eukaryotes differ, right? They have slightly, but importantly, different mechanisms for dealing with this. So in prokaryotes, we have DNA polymerase 1. So DNA polymerase 1 is actually pretty funky. Has two, I don't know, catalytic regions, I guess would be the best way of putting it. Functions, essentially. One is an RNase that recognizes, DNA RNA hybrids, right? So areas of the newly synthesized genome that have RNA in them. And as you'd guess by being an ASE, it's an enzyme that chews up RNA. So that's one function. Second is a short run DNA polymerase. I kind of liken this analogy to like the short stubby trains that move things around in train yards versus like the long distance trains. So DNA pole 1 cannot synthesize long stretches of DNA. Kind of gets tired, falls off, right? It can do short stretches very efficiently though. So essentially what DNA polymerase 1 does is it removes the RNA nucleotide at, you know, the beginning of that primer. And as it does so, it brings in a new DNA nucleotide and adds that on to the 3' end of that fragment. Takes another RNA off, puts another DNA nucleotide on, and it just keeps doing that over and over and over again until it hits the end of, or the beginning, I guess, of the next DNA fragment. And once it's done that, there's no RNA, there's no absence of nucleotides in that strand. But we still haven't finished. We haven't finished because we still have a single-stranded break, a nick. And that has to be filled, right? RNA polymerase cannot do it itself. We need a different enzyme called DNA ligase. And ligate, and this is the same fancy word for the thread that you use to make stitches. It's a ligature, right? Ligate means to stitch together. A little bit of Latin for you. And so ligase, right, is the enzyme, hence the A's, that stitches together the backbone of DNA. And so basically ligase, or DNA ligase, if we want to be specific, plus energy in the form of ATP equals a phosphodiester bond. And we need ATP because if you remember way back here, right, DNA polymerase uses the energy in that triphosphate group, or that phosphate-phosphate bond, to create the phosphodiester bond. Down here, you don't have that, right? So you have to provide that energy separately using ATP. And once that nick has been sealed, to all intents and purposes, you're done. Bar some error checking, proofreading. Does anybody have questions? Yeah. Okay. So actually, this is both prokaryotes and eukaryotes need to do this. What we'll do in a little bit is we'll go through where they differ. And really where they differ, Cynthia, is largely in kind of what enzymes do what. There's a few like little wrinkles there with DNA synthesis and how these primers are removed and replaced. Those are really the main differences in DNA replication between the two. Everything else is largely the same. Okay. So typically it stops when it stops, right? It stops when you're done. Sometimes there are specific termination proteins, right, which block helicase from moving further, and then it's done. But by and large, it's finished when it's finished. Now, it's not actually properly finished yet because we still have another step, right? So our DNA polymerases are pretty accurate, right? And a big part of that accuracy comes from this base pairing or base pair complementarity, right? But they do occasionally make mistakes. Every so often, the wrong nucleotide will come in, and if it manages to stay there for long enough, a phosphodiester bond will form. Now, most DNA polymerases, and they vary, right? Some are more accurate than others, have what's called an exonuclease activity. And exo basically means from the outside, right? Endo from the inside. So you have endonucleases, they'll cut inside a sequence. Exonucleases will remove bases from the end. And so DNA polymerase or DNA pol 3 can essentially stop, right? And what it does is it detects a mismatch typically based on the incorrect size. And when it does, it stops, reverses a base or a nucleotide, and cuts off the offending nucleotide. This is the exonuclease activity. And once that nucleotide, the wrong one, is gone, then it can wait, correct one comes in, and it continues the process. Right, most kind of varies again, but at the very least, DNA polymerases can add between 1,000 and 3,000 bases a minute, give or take. It does vary from species to species as well. However, because this is an exonuclease, it can only cut from the outside. If the DNA polymerase didn't detect that error and it adds the next base, it can't get rid of the wrong base. So it's good at reducing the errors as the DNA polymerase works, but once it's moved on, it can't change that, right? It can only do it for the last base that it added. If it moves on another base or two, essentially it can't go back, even if it's, if it's wrong. So then we have a extra wrinkle. And this is called mismatch repair. So a mismatch or mismatch is a wrong base added by DNA pol 3. And that basically leads to wrong DNA shape. So it's physically, or not visibly, but physically obvious, right? We have a little bubble, right, in our DNA. Now, the issue is, how do you know which is the new DNA, which has the mistake, and which is the old DNA, which does not? right? And this comes down to something called methylation. So the old strand is methylated. And that basically means it has methyl groups added to cytosines. And that process, essentially adding those methyl groups back again, takes time. So the daughter strand is not methylated because it's just been made for a short while after replication finishes. So essentially there's a window of time during which the repair machinery can go, oh, got a problem. Problem is here, and this base here is the one that needs removing. It can only do that while the daughter strand is unmethylated. And the methylases, the DNA methylases, come along fairly quickly afterwards to finish doing the methylation. Now, essentially in the case of an error, there's a whole bunch of proteins, right? And they're all called MUT or MUT. So we've got MUT-S, we've got MUT-L and MUT-H and so on and so forth. What they do is two things. One, they recognize this mismatch based on size. And the second thing is they cut the daughter strand, right, because that's the one that has the error, a little bit further upstream, typically where you next find a methylated A. And those have particular sequences. And so essentially, right, you're not going to be removing just the wrong base. You're going to be removing a bunch of DNA. But it doesn't matter because it's easy enough to replace. And so as soon as that nick occurs, now you have a free end, right? That free end's recognized by a nuclease, and that nuclease will chew up the DNA past the error. And so essentially now you have the same thing as you had before during DNA replication. You have a bit of single-stranded DNA. And so one of the other DNA polymerases will come in and they will fill in the missing nucleotides, and one hopes put the correct one back in, and then seal up the break with DNA ligases. As soon as that's happened, by this point, DNA methylases will come along and they will have methylated that DNA. And now, to all intents and purposes, you no longer have template and daughter or parental and daughter. Now you just have DNA. So essentially, there's a bunch of fidelity, I guess one could call it, right? Accuracy is another way of putting it. Just inherent in the base pairing of nucleotides. And that gives you about at most one error every 10,000 bases, give or take. Just that alone, no other corrective mechanisms will give you a pretty high degree of accuracy. But if you consider that our genome is 6 billion base pairs in total, having an error every 10,000 bases is not going to be good. You'd have to, that would be, so 6 million, that'd be that'd be about 300,000 mutations every DNA replication in humans, right? Which should be terrible, right? Absolutely terrible. So that alone is not enough. On top of that, we have this proofreading function, this exonuclease proofreading function, where the DNA polymerase walks back a base, cuts the offending one out, and then continues back in its job. That will net you a kind of improvement of about 1 in 100. So you'll have 100-fold fewer mutations as a result of this. And then the last ditch or last chance at fixing that problem is with mismatch repair. And that gets you another reduction of about 1,000-fold. And so when you add all of these different accuracy mechanisms together, you get an error between 1 in a billion and 1 in 10 billion. And so that's a big difference. Now, as a human, now you can expect maybe between no and 1 mutation every round of DNA replication, every time your cells undergo mitosis. And that's much, much, much more manageable. That's actually good enough that you won't get all that many mutations incorporated. Obviously you will, right? That's part of how we differ genetically in the food on which natural selection kind of feasts, essentially. Probably change with the age. I don't think so. I don't think the process becomes more error prone with age. And the reason I don't think so is because these proteins are made fresh every time you go through cell division. Right, so they don't hang around. As soon as their cell division finishes, they get broken down and used for spare parts, basically. So I don't think that the error rate changes as we age. There are other pretty fundamental changes that happen as we age, though, and that's down to the linear chromosome problem, the end synthesis problem, which we'll talk about in a little bit. That's actually, in large part, the reason why you get old, which is pretty neat. Sad as well, I guess. Okay, so overall, there's a whole bunch of proteins involved in DNA replication. We've got our gyrase, we have helicase, we have our single-stranded binding proteins, we have primase, DNA polymerase complex, other DNA polymerase involved in removing the primers and synthesizing DNA, and DNA ligase. Right, so this is a lot of stuff to remember. The best way to remember it, for me, hopefully for you too, but not necessarily, is to understand what these different things do. Right, what is their role? Understand that, and it becomes a lot easier to remember what those different things do, where they act at, things like that. Okay, any questions? Oh, and kind of getting back to Michael's question about probability of error changing with age. Another one thing that does affect the number of errors you get every round of replication is any mutations in the DNA repair machinery, or the proteins that recognize when chromosomes are broken or incompletely replicated. So genes like BRCA1 and BRCA2, for example, that are breast cancer risk factors, their main role is in DNA repair. So essentially, when they don't work, you don't catch these errors, and those errors accumulate to the point where they may affect something important in the cell cycle or cell cycle control. Okay, so by and large, DNA replication in eukaryotes is pretty similar. But the differences come about largely from the structure of the chromosome and how DNA exists in a cell, right? So eukaryotic chromosomes are really huge compared to prokaryotic genomes. They're linear, which creates big issues later on and is the reason why you get old. And they also have proteins called histones attached to them, basically to form chromatin, which is protein and DNA. And so what I'm going to do next, well, I'll see how I get on. See if we have time. I'm kind of starting to run out of juice, to be honest. Actually, you know what? I might call it a day today. I'm just going to leave it here with chromatin. We'll come back to this again later on because it's a really important part in gene expression. And so basically in eukaryotes, chromatin solves the problem of fitting lots of DNA into a very small space. And so it doesn't matter in prokaryotes because they don't have nuclei, right? They don't have a nucleus, right? Their DNA is sort of localized to one part of the cell, but it has the whole cell to hang out in. Eukaryotes don't have that luxury because all their DNA is packed into a really small part of the cell called the nucleus. Right, really small as in like the cell's small, nucleus is smaller than that. And so eukaryotic DNA is aggregated or wrapped around other proteins. Those are called histones, right? And aside from the whole deal of how that affects gene expression, which we'll cover in a lot more depth later, you have to get these off, right? If RNA or DNA polymerase is able, well, RNA polymerase too, to copy that DNA. And also, you got to put them back on again, right? And so every time you synthesize DNA, you have to, as a eukaryote, you have to remove these chromatins and make exactly twice as many as you had before. The same amount again. Because all of that new DNA is going to need histones to be wrapped around, right? So there's actually an awful lot of proteins that have to be synthesized for DNA replication in eukaryotes. And just as a kind of an interesting aside, right? So, and just as a like fun thing to end up the class with, I guess. So you've probably been taught quite correctly, right? That all of your DNA comes from all of the information in your DNA, right, is in the base or form of your DNA sequence, right? Series of DNA bases equals genes or promoters or whatever, right? And that's what you inherit from your parents. Well, that's not actually 100% correct. What you also inherit are some of the chromatin marks. And so these are marks which do not change the DNA sequence, but do change how that DNA sequence is accessed. And these are marks that you add over time yourself to your DNA and to the proteins your DNA is wrapped around. So a quick aside, I don't know if anybody has twins in the family. Twins are a great example of this. Identical twins, that is. Or maybe you're even an identical twin yourself. One of the students I worked with over the summer is an identical twin. Her sister's, I think, in Pakistan. No, Bangladesh, that's where she's from. Right. So if you look at identical twins as they're younger, particularly if their parents dressed in the same, which I always find a little strange, but they will look pretty much identical. Look at that same pair of twins and they're in their 50s, they will look almost completely identical. But you will be able to differentiate between them. That difference is based on the chromatin marks that they accumulate. So over time, how their genes get expressed, even though they have exactly the same DNA, right? They are identical twins. How that DNA gets expressed is going to change ever so subtly, and you're going to be able to see that. So identical twins in middle and late ages are not absolutely identical. And so some of those marks you pass down to your children. So essentially and this is much, much more important for the matrilineal line, like the female line, than it is for the male line. It still applies to males, but essentially part of who you are as individuals, not necessarily a big part, but a part nonetheless, is based on the experiences, right? The environment that your parents experienced, particularly your mother, before you were born. Isn't that crazy? I always think that's fun to talk about. Anyway, that's enough from me. I'm starting to slow down. It's like a record player that's run out of batteries. Have a great weekend. Next week, we'll finish off eukaryotic DNA replication, like those differences. Do we talk about the end replication problem? Ah, yes, we do. That's always fun to talk about, i.e. what makes you old. And then we'll talk about homologous recombination. If we have time, which we may do, hopefully, bring any questions you have and we'll go through that in class. All right. Have a fabulous weekend, everybody. See you next week.